relation between nucleotide excision repair and cancer. So first, the mechanism. Well, in the previous talks, you already heard about other DNA repair pathways like mismatch repair and also base excision repair. I'm going to talk about nucleotide excision repair, and it's a related pathway to the base excision repair pathway. So like base excision repair, nucleotide excision repair deals with lesions that affect a single strand of the DNA, but because of this, they disrupt the double helix in such a way that these lesions are being recognized by nucleotide excision repair. So examples of such lesions, it's already mentioned, are lesions, for instance, introduced by UV light from the sun. This would be UVB and UVA light. In the lab, we also use UVC light, which is more reactive to DNA. But fortunately, we are shielded from that type of UV irradiation by the ozone layer. And what UV light does is that it can introduce different crosslinks in the DNA. And the most frequent crosslinks are called cyclobutane pyrimidine dimers, which is a crosslink between two adjacent pyrimidines via two carbon atoms, or a bit less frequent but more mutagenic are the 6,4-pyrimidine pyrimidone photoproducts, which is also a crosslink between two adjacent pyrimidines, but then involving one carbon of each pyrimidine. These are called UV photolesions, and we use them a lot in the lab as a model to study how nucleotide excision repair works, because you can imagine that it's easy with a UV lamp to rapidly induce lots of DNA damage such that you can study the cellular response to this. But there's also other types of damage that are being recognized by nucleotide excision repair. So for instance, other bulky adducts like monoadducts, for instance, from compounds that are found in smoke or in your favorite meat that you grill on the barbecue, you can find compounds that interact with the DNA and create this helical distortion. And in a similar manner, certain chemotherapeutic drugs like cisplatin, oxaliplatin, carboplatin, they also react with the DNA. They create some interstrand crosslinks, so between the two DNA strands, but also a lot, even much more, intrastrand crosslinks, which would be crosslinks between two adjacent bases or even bases a bit further apart, which also induce a helical distortion, and that's being recognized by nucleotide excision repair. So the nice thing, for us at least, is that nucleotide excision repair can recognize such a large variety of lesions. And this is the case because they all induce this helical distortion. So it's not really the lesion itself that's recognized by nucleotide excision repair, but more the effect of the lesion, as I will show you in the next slides. So nucleotide excision repair basically consists of two sub-pathways, one which is called the global genome nucleotide excision repair pathway, and the other one is transcription-coupled nucleotide excision repair. And the global genome nucleotide excision repair pathway is initiated anywhere in the genome by this XPC protein complex, which just freely diffuses through the nucleus, binding to DNA and looking whether there's any damage there or not. And whenever it finds the damage, it binds more tightly to the DNA and then initiates nucleotide excision repair. The other pathway is the transcription-coupled repair pathway, which is initiated by basically stalling of RNA polymerase II. So RNA polymerase II is transcribing a gene, and when there is a damage in this transcribed strand, it will stall on that damage. And then there's another protein called CSB, which tries to push RNA polymerase over the damage, because there's also natural pause sites, for instance, where RNA polymerase II can stall. If CSB can push RNA polymerase further, then transcription just resumes. And if CSB cannot do this, CSB binds more tightly, and that also initiates then transcription-coupled nucleotide excision repair. Now I'm going to talk about the mechanism of both these repair pathways, which basically share the same mechanism in which they take away or remove the damage. And this happens in different steps. And you can really divide nucleotide excision repair very broadly in 4 different steps, which I'm going to do also for simplicity. But of course, you should realize that these steps, they are interwoven. And one protein of one step can also reciprocally interact with proteins of the previous step. And what is really nice about nucleotide excision repair is that you can see that proteins also mutually stabilize each other and each other's binding to the DNA. And I will give you some examples of that. So global genome repair is initiated by this damage recognition step, which would then be the first step, which is initiated by the XPC protein. Actually, this is a protein that always exists in a trimeric complex together with RAD23 and centrin-2, which also stabilize XPC. And as I already said, what XPC does, it just floats around in the nucleus, binding DNA, looking for any damage. And the way it does this is through different domains. So this is a structure of yeast XPC protein that they solved a bit more than 10 years ago, and that showed us how XPC actually detects DNA damage. And it does this by binding with two domains, the TGD domain and the BHD1 domain. It binds to the double-stranded DNA, and it does so in a transient way, so it constantly binds and then lets go. So it's a very dynamic process. And at the same time, then with another domain, or actually two domains, the BHD2 domain and the BHD3 domain, the BHD3 forms this hairpin domain. It tries to insert these domains in the double helix. Well, normally this cannot happen because the double helix, of course, forms a tight helix. But if there's a damage there, then there's a helical distortion, and then XPC, if the energetic barrier is low enough, it can insert this BHD3 domain in between the double helix and then bind more tightly to the DNA, which is the trigger to initiate nucleotide excision repair. And in this way, this also explains this binding mode of XPC. This also explains why XPC can recognize such a large variety of different types of damage. So that's really different from the base excision repair pathway that you heard in the previous talk, which has all these glycosylases that are kind of specific. Of course, there's a lot of redundancy, as we heard. They're kind of specific for specific types of damage. XPC can just detect a helical distortion created by many different types of damage. Now, not all different types of damage cause sufficient helical distortion for XPC to bind tightly to the DNA. And therefore, there's also another protein complex, which is called the UV-DDB complex, consisting of DDB2 and other proteins that together form an E3 ubiquitin ligase complex. And this DDB2 protein, it can recognize some lesions very specifically and also bind to these lesions. Like CPDs, it can bind these, and CPDs do not distort the double helix very much, such that XPC can easily recognize them. But by binding two CPDs of this UV-DDB complex, it also flips out the damage and also creates a more suitable substrate for XPC to bind there. So the UV-DDB complex helps XPC find damage. And with this E3 ligase complex, it then also ubiquitinates probably many target proteins, although we don't know which exact targets are there. But for instance, it ubiquitinates itself, and with this, it regulates its own function. And also it ubiquitinates XPC, and by doing that, it likely regulates also the affinity that XPC has for DNA damage. And finally, this UV-DDB complex is also thought to regulate chromatin remodeling, because you can imagine if DNA repair has to take place within a chromatinized DNA, that the repair reaction cannot always easily take place because maybe nucleosomes are in the way, and these nucleosomes also need to be removed or remodeled in order for repair to take place efficiently. And it's also thought that this complex plays a role in that. So this is the first step. And once XPC binds then tightly to the lesion, it recruits the proteins that execute the next step, which would be the TF2H complex, XPA protein, and also the RPA protein. And the TF2H complex is a 10 subunit complex. It stands for transcription factor 2H. So TF2H also acts in transcription. It's actually a transcription initiation factor that helps loading of RNA polymerase II on the promoter. And it does so via a helicase subunit, the XBB helicase subunit, which is important for transcription, but less important for nucleotide excision repair, because in nucleotide excision repair, it's especially the XPD helicase, which is a 5' to 3' helicase that is important. And with this helicase, the TF2H complex unwinds the DNA, so creating a larger bubble. And in that way, by doing this, it also searches for damage, because XPD has this hole through which it pulls the DNA while doing the unwinding of the DNA. If there is then a damage on the DNA, this activity is stopped. And in this way, the TF2H complex verifies, checks whether there's really damage or not. Because XPC recognizes helical distortions, maybe it also recognizes the helical distortions not induced by specifically DNA damage, but maybe, for instance, a mismatch, and then nucleotide excision repair does not per se need to take place. So this is an extra verification step to make sure there's really damage there. And the TF2H complex is being held by the XPA protein in this function. So in this way, the idea is that there's a tripartite damage recognition step in nucleotide excision repair, in which first XPC searches the DNA looking for any damage, which you can see in this animation. It flips out the damage then, and then comes the second complex, the TF2H complex, together with XPA, which then scans along the DNA until it finds the damage. And this really stabilizes this, what we call the pre-initiation complex, also together with this RPA protein complex, which you might know, that's a single-stranded DNA binding protein complex. And that binds to the opposite DNA strand, opposite of the lesion, thereby also stabilizing this whole complex. And together with XPA and RPA, they make also sure that the next step can take place, which is recruitment of two endonucleases, the XPG endonuclease and the ERCC1 XPF endonuclease, which incise the DNA around the damage. So that is then the next step, which is the damage excision step, executed, as I said, by XPG and ERCC1 XPF. So the XPG protein, well, there's a bit debate in the field whether that's already part of this TF2H complex or not, or whether it's recruited really separately. Anyway, this XPG protein is thought to be needed to be present here before the ERCC1 XPF complex can then first cut on this side of the damage. So first the cut is made here at the 5' side of the damage. And after that, XPG cuts here at the 3' side. And this leads to the excision of a strand of 22 to 25 nucleotides containing the damage. And this is also the reason why this repair pathway is called nucleotide excision repair. Now this leaves a gap, which is basically a single strand break. And it's also seen by the cell as a single strand break because of this also DNA damage signaling occurs. So for instance, there's activation of ATR, phosphorylation of H2X, so formation of gamma H2X, ubictylation of histones, and also DNA damage signaling by well-known damage signaling factors that are also found in double-strand break repair, for instance, or at the stalled replication forks, like RNF8 and 53BP1. I'm not going to go into this, but there is lots of DNA damage signaling here, which is often not realized. For instance, if people look at gamma H2X, they don't realize that this can also happen at these single-strand breaks. And the exact function of this DNA damage signaling in nucleotide excision repair is also not really known. Maybe this has to do with regulating the cell cycle. So we get this single-strand gap, and that then needs to be filled up. So that's the final step of nucleotide excision repair, the gap filling step, which is executed by proteins that are also involved in replication, like we already saw them in previous talk, like the RFC complex, pCNA trimeric complex, which is needed to load DNA polymerases, and in nucleotide excision repair, both DNA polymerases delta and kappa together, or DNA polymerase epsilon is implicated in filling this gap, and which is then finally ligated by either ligase 1 or ligase 3 complex, probably depending on the cell cycle in which this takes place. So this is basically the global genome nucleotide excision repair mechanism. The nice thing about this last step is that you can use it in the lab also to measure whether the global genome repair is active. And of course, in patients, you can then also use this as a diagnostic marker, because you can incubate cells with a thymidine analog, ethylene deoxyuridine, so EDU. You can UV irradiate cells and then just measure by making this EDU fluorescent. You can just see under a microscope, and then also measure by measuring the intensity whether nucleotide excision repair takes place, as in this control sample, or not, if you, for instance, knock down the XPC protein. This is global genome nucleotide excision repair. The other pathway is the transcription-coupled repair pathway, and I already said that the basic core NAIR machinery of transcription-coupled nucleotide excision repair is the same as global genome repair. The only difference is the initiation. So transcription-coupled repair is coupled to transcription because it's initiated when RNA polymerase II stalls on a lesion. And when then the CSB protein cannot push RNA polymerase II further over the lesion, because of this, binds more tightly to RNA polymerase II, and then recruits the subsequent TC NAIR factors, which are the CSA protein and the UVSSA and USB7 protein. So the CSA protein forms a complex, kind of similar as the DDB2 protein in global genome nucleotide excision repair. It's the same complex. The only difference is that here there's CSA, and in the other complex there's DDB2. So this is an E3 ubiquitin ligase complex. And this complex is being recruited to CSB and then also starts ubiquitinating target proteins. And also in this case, it's not really known which exact proteins are ubiquitinated. In the Netherlands, there's labs investigating this, but it's likely that CSB is ubiquitinated by this complex, and also RNA polymerase II itself. And this ubiquitination of CSB leads to the degradation of CSB and also makes CSB unstable. Fortunately for CSB, then this other complex is also recruited, which includes the USP7 protein, which is a deubiquitinating enzyme, and this counteracts this ubiquitination and deubiquitinates CSB, thereby stabilizing CSB also. So this is such an example of a downstream protein that reciprocally interacts with an upstream protein and stabilizes its binding, also its activity at the site of damage. The other protein that's being recruited is the UVSSA protein, and UVSSA, similar as XPC, is involved in the recruitment of TF2H, which initiates the next step of nucleotide excision repair. So which is the damage verification step, and which is done by the TF2H complex together with XPA again, which then leads to recruitment of the endonucleases, ESCC1, XPF, and XPG, which inside the DNA leads to the excision of the damaged strand. And then finally, this gap is filled by polymerases. So that's the gap filling step. Now we think that this this downstream mechanism of transcription-coupled repair is the same as in global genome repair. It's not really sure, because you can, for instance, think that this damage verification step, in which TF2H is involved, may not be necessary for transcription-coupled repair, because already when RNA polymerase II stalls on a lesion, that basically means that there's a lesion there, so it does not need to be verified. So the idea is that maybe TF2H has a different role in this pathway, and that's through its unwinding activity of the DNA, so opening up the DNA bubble, making the transcription bubble bigger. Maybe it's involved in reverse translocating RNA polymerase II, so moving it away from the damage, so there is space for the whole nucleotide excision repair reaction to take place. That is currently under investigation. Now, transcription-coupled repair, you can also easily measure in the lab whether it takes place or not, and we don't do that by EDU labeling. Well, we can, but there's also another technique by which we do that, and that is by incubating the cells with a uridine analog, EU, ethynyl uridine, which is built into RNA, because if you UV irradiate the cells, because RNA polymerase II stalls on DNA damage induced by UV irradiation, you also block transcription, which you then can measure if you visualize transcription by EU labeling. So this would be an undamaged cell. This would be 2 hours after UV irradiation. And in time, you can see transcription coming back again, if there is transcription-coupled repair. If there's not, then you don't see this happening. So this is a relatively easy way in which you can measure the activity of this pathway. So this is basically the or the two nucleotide excision repair mechanisms. So what does this have to do with cancer? That's what I want to discuss next with you. So nucleotide excision repair and cancer. And then specifically, I want to talk about nucleotide excision repair disorders. So inherited deficiency in nucleotide excision repair. So if you think of nucleotide excision repair and cancer, actually, so there's two negative side effects of DNA damage accumulation. So if DNA damage is not properly removed, one we focused on already also, of course, in the previous talks, which is cancer. And if you think of nucleotide excision repair, then for instance, you think of cancer induced by mutations induced by UV damage. So that would be, for instance, melanoma. If you look at the incidence of melanoma in the Netherlands, you also find that this is the most frequent type of cancer. For instance, caused by UV damage. So that also shows that nucleotide excision repair must be a really important pathway to prevent cancer. And the other side of DNA damage is aging, which is very well or very nicely exemplified by this image. Maybe you've seen it before. So this is a famous image in the field of a truck driver who was driving his truck for, I think, decades, with always the window on his left side open. So the left side of his face would be exposed to the sun, to UV light, whereas the right side was inside the car and that would be kind of shielded. And you can see the enormous difference in if you think of skin aging, if you look on the left side of the face and the right side of the face. So this very well exemplifies that DNA damage can not only cause cancer, mutagenesis, which then leads to cancer, but also cause aging. And those two sides of the DNA damage coin that you also see in nucleotide excision repair deficiency disorders. So cancer, you see, for instance, if you look at deficiency of this global genome repair, nucleotide excision repair pathway, and aging, you see if you look at inherited deficiency of the transcription-coupled nucleotide excision repair pathway. So deficiencies in global genome nucleotide excision repair lead to the syndrome that we call xeroderma pigmentosum, and deficiencies in transcription-coupled nucleotide excision repair are associated with another syndrome called cocaine syndrome. So first, xeroderma pigmentosum. So that's the typical disease that is diagnosed at an early age. Parents that take their child, for instance, to the beach, and a child is for the first time exposed to UV light from the sun and immediately burns. So within minutes or within 50 minutes, the child gets these very severe sunburns on the skin. The parents take the child to the doctor and xeroderma pigmentosum is diagnosed. These children also often get at a very young age different types of skin cancers and also other types of cancers. And the reason for that is that they have defects in the global genome. I hear the sound of somebody with papers. And maybe everybody turn off their sound. I don't think the person hears me. Anyway, so this is typically caused by mutations in genes involved in global genome repair. So like the XPC protein, the XPA, and also other proteins. And this also immediately explains why these proteins are called XP, whatever, because XP stands for xeroderma pigmentosum. So these genes were also originally identified and cloned in xeroderma pigmentosum patient cells. And this syndrome is then characterized by extreme sun sensitivity and also an enormous increased risk of developing skin cancer and also other types of cancer. And also the more severe patients, also they develop in later life, they develop progressive neurodegeneration. So which means that their nervous system starts to functionally decline in a progressive manner, meaning that it gets worse with age. And that's also because it's caused by DNA damage. So it's DNA damage accumulating, and because of that, mutagenesis occurring, more mutagenesis, and because of that, cells also deteriorating and also developing cancer. Now, there's no treatment for this syndrome, and the only way in which this can be managed, or the best way, is just by avoiding anything that causes DNA damage, like avoiding the sun by wearing protective clothing, or even avoiding going outside at all during daytime. This is xeroderma pigmentosum. So the other syndrome is cocaine syndrome, which is a totally different syndrome. It's not characterized by cancer development. On the contrary, often these patients do not develop cancers, but they show developmental problems. Growth failure, which can either be at a very early age or develop a bit later in life, depending on how severe the disease is. They show growth failure. They also show progressive neurological problems, like an intellectual decline. And very strikingly, they show this premature aging phenotype, which is well shown by a famous patient in our field, who was called Baptiste. He lived from 1993 to 2003. And as you can see, at a very early age, he looked like a normal developing boy. But as he got older, there was a gradual functional decline until he reached the age of 10. And here you can see him in a picture with his sister, who is only 1 year older. And you can see that he basically looks like an old man. So this is what we call accelerated aging, which means that already during childhood, you see these aging features in these patients. And this is caused by defects in two nucleotide excision repair genes, which are involved in transcription-coupled repair. So the CSA and the CSB gene. So CS also stands for cocaine syndrome, as you now understand. So these are the most well-known syndromes associated with nucleotide excision repair deficiency. And that also shows the extreme importance of nucleotide excision repair, because if you do not have functional nucleotide excision repair, you have these very devastating types of disease. There's also other types of disease associated with nucleotide excision repair deficiency. So not only xeroderma pigmentosum or cocaine syndrome, but also combined forms of this disease. So patients that have a mutation in a gene, downstream gene, that functions in both pathways, have this combined xeroderma pigmentosum cocaine syndrome complex. There's also another disease called trichothiodystrophy dystrophy, which can also be combined with xeroderma pigmentosum. And this is also caused by mutations in their genes, but especially genes that are part of this TF2H complex. And likely these mutations affect more the transcription function of TF2H than the nucleotide excision repair function per se. And therefore, this disease is also thought to be more a disease of transcription problems than nucleotide excision repair problems. And finally, and that's kind of intriguing, there's another syndrome which is called UV sensitive syndrome, and it's really rare. And this is also caused by a mutation in the transcription coupled nucleotide excision repair protein, so the UVSSA protein. But strangely, this does not cause the very severe cocaine syndrome, but a very mild syndrome in which patients are only mildly UV sensitive. And this likely hints to the fact that it's not per se a defect in transcription coupled nucleotide excision repair that's the problem in these patients, but it's more the problem of what happens to RNA polymerase II. The model that we in our lab favor is that in cocaine syndrome, RNA polymerase II stays on the lesion, and because of that, the lesion cannot be repaired. So, and that forms a bigger problem than in case of the UV sensitive syndrome. The lesion is also not repaired, but RNA polymerase II can be removed from the lesion. And maybe in that sense, the lesion can be repaired in another way, or maybe cells are less sensitive to that. So that brings me not yet to my last slide, but this slide to show you that there's a lot of things. So I explained to you the nucleotide excision repair mechanism and also the main syndromes associated with its deficiency, and also showing to you that the nucleotide excision repair is therefore very important to life and also to protect us against cancer and aging. But there's also a lot of things that we do not still know about nucleotide excision repair, especially about its mechanism. There's a lot of research still going on. One of the intriguing questions is, for instance, how it can be that different mutations in these genes can give rise to these different syndromes. So not only xeroderma pigmentosum or cocaine syndrome, but also this, for instance, this very mild UV sensitive syndrome. And besides this, there besides that, there's many other questions, and I'm not going to go into all these questions. This is just to indicate to you that there's still a lot that we don't know. So what I find personally interesting, and also in light of the theme of this course, is the relation of nucleotide excision repair with cancer, but not only with cancer, but also with cancer therapy. Because, as I said in the beginning, so nucleotide excision repair can detect many different types of damage, also those induced by platinum drugs like cisplatin, carboplatin, oxaliplatin. And in that sense, nucleotide excision repair not only protects against cancer, but it also protects cancer cells against cancer therapy, because this is a very widely used and applied chemotherapeutic drug. So in that sense, it's interesting to think about whether we can use the knowledge that we have of nucleotide excision repair to improve cancer therapy. So maybe you can think of maybe there's nucleotide excision repair deficiencies in cells that we can exploit to make cells more vulnerable to these kind of drugs. Or maybe nucleotide excision repair is involved in resistance of cancer cells to these drugs. So that's a thing that I'm interested in. And also another thing is that a problem with these chemotherapeutic drugs is that they have a lot of side effects, and they especially affect the peripheral nervous system. So maybe you know of people that have had chemotherapy, you know that these people get tingling sensations in their fingers, for instance. And that's because these drugs are very toxic for neurons, for peripheral postmitotic neurons. And we were also interested in finding out whether maybe nucleotide excision repair has something to do with that. So I just want to give you a very short idea of what we do in our lab with the relation between nucleotide excision repair and cancer therapy. And that is that we are interested in the response of cells to oxaliplatin. And oxaliplatin is the type of cancer drug that is used to treat colorectal cancer. Because for some reason, colorectal cancer cells, they respond better to oxaliplatin than to cisplatin. So which are very similar drugs. So the big question is, why is this? Why do certain cells respond better to one drug than to the other drug? And we thought, well, maybe nucleotide excision repair has something to do with that. So to start studying this, what we did in our lab, we created a CRISPR-Cas9 library to knock out many different DNA repair genes, creating like 80 cell lines, having a deficiency in a specific type of DNA repair gene. And we exposed these cells to oxaliplatin just to see which DNA repair pathway is now most important to protect against oxaliplatin treatment. So the results from 2 of those screens you can see in this graph. So this is screen 1, this is screen 2. The cells that are here are most sensitive to oxaliplatin. And what we found is that when you knock out genes for specific DNA, for different DNA repair pathways, like the Fanconi anemia repair pathway, molecular recombination, translesion synthesis, you sensitize cells to oxaliplatin, and this was also as expected. But to our surprise, we also found among these top hits, we also find nucleotide excision repair and even base excision repair genes. I'm not going to go into the base excision repair, but for us, it was surprising that if you look at the nucleotide excision repair genes, that we found CSA there, and not, for instance, XPC or DDP2, which were involved in global genome repair. So to us, this shows that it's especially the transcription-coupled nucleotide excision repair that is important to protect cancer cells against the effects of oxaliplatin. So that's a thing that we're currently investigating in the lab, where there may be differences in whether global genome repair or transcription-coupled repair is involved in cells in protecting against oxaliplatin or cisplatin, because we see differences between these drugs when you look at global genome or transcription-coupled repair involvement, whether that may explain why certain cancer cells are more sensitive to one drug compared to the other drug. And because of this importance of the transcription-coupled repair pathway, we also think that maybe the severe neurotoxic side effects of these drugs are caused by specific interference with transcription in, for instance, neurons.
The laptop.
Neurons very much depend on transcription integrity. So this is still ongoing. Maybe in the next time or at a meeting, whenever I may meet one of you, I might give you more details about our ongoing research. With this, I would like to end this talk. If you want to read further about nucleotide excision repair, you can look in any of these papers, which are from our department, but of course, also other interesting papers. This is just a selection from us. And finally, this paper describes our findings on oxaliplatin and transcription coupled and base excision repair. So thank you for your time. And if you have any